So welcome to today's webinar. We'll be demonstrating the integration between LabKey Server and FCS Express flow and image cytometry software from Novo software. I'd like to take a moment to introduce ourselves. My name's Kaya Ghosh. I'm a technical application specialist with Novo software, the makers of FCS Express. FCS Express is a full-service cytometry data analysis and reporting package. We can read FCS files from any commercially available cytometer, as well as many image and image cytometry data formats. With us on the line is Bernie Lee from LabKey Software. Since 2013, Novo software has worked with our partners at LabKey to become fully compatible with their import process via our normal batch processing functionality, which is probably familiar to those of you who are already using FCS Express. Bernie is going to be showing us around within LabKey to show you all the sophisticated data reporting tools that are available to you. And we'll be doing that portion of the webinar right after we show you how the export works from FCS Express first. So just a little bit about LabKey Server. LabKey is an open source platform for scientific data management and analysis. LabKey focuses on development of tools needed to facilitate biomedical advancement. In that effort, LabKey provides the ability to upload from lots of data sources, including assays, instruments, clinical databases, specimen data, etc., and integrates the information for analysis, visualization, and publication. LabKey provides a rich security and permissions model for securely sharing your lab data with collaborators in a read-only fashion. So if you can all see my screen, I think what we can do is just go ahead and get started and show you how the whole export-import process works from FCS Express into LabKey. But before I do that, I just wanted to point out that we do have tutorials available from within FCS Express and on the web. All you would need to do is come to File, Help, and click on Tutorials. And then if you just search for LabKey here, you'll have the LabKey tutorial coming up. And within that tutorial, it links out to the twin tutorial on the LabKey side that shows you how to actually access your data once you're in LabKey. So just feel free to kind of watch and get an overall feel for the process because the individual steps are enumerated within those tutorials. Okay, so here is a very simple layout. And in this layout, we're going to export a few different statistical tokens, keyword tokens, which if those of you, some of you may not be familiar with keywords, but keywords is basically metadata that's with your FCS files. An example of that is the date. So all of your FCS files will have a date keyword so you know when it was acquired. And we'll even export some additional file types like pictures and raw FCS data files to LabKey. Okay, so for those of you who aren't familiar with FCS Express, just to show you a basic idea of how it works, this is an experiment with 7 different data files. And I can just click Next and Previous and see that the layout essentially functions as a template through which we cycle our data files. And what we're going to do is export this complement of data from all of the data files to LabKey. And like I mentioned earlier, that works via the batch processing module in FCS Express. So if you come over to the batch tab, and in case you haven't noticed, we model our software on a Microsoft PowerPoint-like interface, so it should be easy to find everything. So the batch processing is here on the batch tab. So I'll just open up my batch actions, and you can see that we have a few exports already lined up here. So we're going to be exporting the metadata of participant ID, date keyword, which I mentioned earlier, the percent of gated cells in this M1 marker, and we're even going to export the full FCS Express layout, the raw FCS file data, and one of the pictures. So all of these plots here on the layout are individual objects which can be exported as pictures of any format. We're going to go ahead and export a JPEG. And let's put in one more export just so I show you how easy it is to set up these exports in FCS Express. We'll put in this one here, the number of events in gate 1. So I'll drag that over. And now we just need to come into that export, and under field, we need to type in the XML node. And so the XML node corresponds to the name of the data field within the LabKey assay design that you've established. And Bernie will show you a little bit more about that in his portion of the webinar, just how you define an appropriate assay design for your needs within LabKey. Before we kick off this export, I'm going to click on the action, which is, by the way, from here, add report. We've already added in a LabKey action. And the LabKey server URL and protocol or assay ID here in options, that's also accessible from within the import data section of the LabKey assay design. And our tutorial will tell you how to get that information. We have also filled out our credentials in advance here, and these are the credentials as defined by your LabKey server account that you need to export to the assay design. We'll also give this particular run a name. So I'll call this today's date, and we'll put in a comment. So this comment can be anything you want it to be. It's actually not mandatory, but I like to add it. I think it's a useful descriptor when you're coming back and looking at your multiple runs exported to LabKey. So now I'm going to click Run, which is here on the Batch tab. And now you see we have a batch processing window popping up, and it's going through and telling us about the progress of all of the iterations that are being exported to LabKey. So it looks like it's just taking a minute to finalize the export here. And again, if there's any questions at this point, please enter them into the questions and answer box. We will certainly be happy to address those as we go. So now I'm going to come to LabKey, which is a very convenient web interface. It's really easy to get to your results here. And I'm here in my list of assays. We have a few different assay lists defined here under this test case account. So we're interested in the webinar assay list that we've just exported to. And I click on it, and you can basically see, let me try and highlight the timestamp here. So this is the data that we've just exported here at 10:07 a.m. And what we're going to do is click on it and take a look at the actual data that was exported. Okay, so you see we basically have one column of data for every item that we exported. These first two columns on the left here are keyword metadata that was saved with the FCS files. Then we have a couple of statistical tokens here in columns 3 and 4, the events and the percent gated within that marker. And we even have the FCS Express layouts, FEY, which can be archived within LabKey just like any other data file format, the raw FCS data files, and we have these JPEGs that we exported, which are pictures of the histogram from our plot. And you can mouse over them and see a little preview of how they look. So hopefully I've shown you how easy it is to set up those exports and view them within LabKey. If there's any questions, like I said, just go ahead and enter them in. And at this point, I am going to switch over the presentership to our partner at LabKey, Bernie Lee, and he's going to introduce himself and show you around within LabKey.
Hello, everyone. As Kaya said, my name is Bernie Lee. I'm a program manager for LabKey Software. And today, I would like to briefly walk you through some of the features that Kaya described about LabKey. One of the fundamental components of LabKey is the assay and assay design. Assay allows you to store data and group data by batch, run, and down to the individual data level. Now, when preparing for to receive any kind of export, we first create a new assay. And as you can see, LabKey offers a number of prepackaged assay designs, in this case, including an FCS Express design. You can see some of the other options here. Now, we are taken to the assay design screen after selecting an assay type, where we get to fill out information about the assay. In this case, all of the fields that we actually are interested in collecting. So we have the batch field information, which captures metadata about a reasoned grouping of assay runs. In this case, the specific cytometer, machine operator, or which grant is actually funding this particular work. You can also capture run field information, which is metadata about a given experiment, the cell type, the date, type information. And then down to the individual run itself. These data fields capture experimental result types and names. Now, as you can see here, there are a couple of pre-populated fields. The ones here are for use in LabKey's optional study framework, which I will touch on a little bit later. But of course, in our case, we're going to want to add some additional fields, which is very simple. You can add the field here, and in this case, we might be interested in those Gate 1 events, which we can title. And then we get to choose our data type. LabKey offers a number of options. In this case, of course, we're counting in individual cells, and we're interested in integers. But you can set it as a string type, date type, as well as the files that, such as the histogram that Kaya showed before. And you simply populate each of these fields in the way that the export will show up, and the files, including protocols or images, will show up as thumbnails. Once these are populated, normally you'd simply save and close, and your new assay design would appear. But in order to save a little bit of time, I'd like to just show you one that I previously made to match the export that Kaya produced. The main things that I've added are these 4 fields at the bottom, including our events integer, as well as the JPEG that she showed, the FCS file, and layouts in file format. Now, once you've produced your assay design, LabKey is prepared to receive and store all the data we are interested in. One simply exports the run in the way that Kaya demonstrated, and we're ready to go. Now, after export, you click on your file. Just as the way that Kaya showed, you'll now get this data grid, and we are prepared to perform some sort of analysis or to create charts or reports. The fundamental data interaction feature of LabKey is this grid view that you see before you. It supports sorting, multifaceted filtering, as well as the creation of quick charts. Now, we have the ability of creating custom views, and in fact, you'll notice that there are a couple of columns already here that were not part of the original export, such as this comments field, which Kay mentioned we could create. This grid also supports exporting. If you'd like to view your data in Excel or in some text format, including TSV or CSV. And you can also access the data from an external tool through APIs by exporting this script into your favorite programming language. Now, this data can be charted in many forms. In the most basic form, we simply select a data value column and choose the quick chart option. And LabKey will predict what type of chart you're interested in viewing. Now, in this case, we selected only a single column, and so it predicts that we'd like to see a box plot of the 7 data points that we created. And there you see the predicted quartiles, max, and min information. But we might be interested in something different. So in this case, we will come and we will change our x-axis, indicating that we want to see this information over time. And LabKey will predict that we now want a scatter plot, which with 7 data points doesn't necessarily show you a lot, but you can do some additional distinguishing, in this case, say, of the participants involved in this experiment. So I'd like to show you how to group. In this case, we're going to group by the participant ID. That way we can distinguish between the people involved. When we do this, we now have 2 distinct participants. Then we can save the reports for quick viewing, as shown here. I will show you a previously created quick view in a few moments. And you can also export these plots in exactly the same way that you could export the data grids. In this case, as a PDF or as a script to embed in something else. Next, I'd like to show you a little bit about our study, the study functionality. So a study, a LabKey study, tracks and integrates information through time. It will allow you to combine your assay data with other data sets, such as specimen data, physical exam or demographics data, and any other assay data that you would like to integrate through the multiple time points used in lots of longitudinal research studies. So first, what we have to do is copy our assay data into that study. In this case, I'm going to select all of it, and you can simply press this Copy to Study button. However, we might want to be selective about the data we choose, should any of our data fail the quality control testing that each of us perform. So we can simply uncheck what we don't want. But in this case, I'd like to copy all of it in. So we press Copy to Study, which essentially just creates a pointer to the existing data, and we choose where we'd like to copy it. Now, I have a number of studies set up, but I'd like to do this HIV CD4 study. When we press Next, we get a preview of what our data will look like and how it will be integrated. It shows us the 2 participants that are involved, as well as various date information, the time points in which we took this data, and how that time point translates into our study, how it's going to be integrated. So we know where it will end up. So we copy to study if we don't want to change anything. And now it says we are in this interactive study. Now I wanted to show you a little bit about study generally. So here is our study where we are comparing antiretroviral treatments. And you'll notice that there are a number of data sets down here, including now our FCS protocol, which we just exported. And there are a number of other data types, including this physical exam, basic test results, consent information, as well as a number of assay options, including Luminex, ELISpot, neutralizing antibody assays, microarray assays, and many, many more. Now, we get some additional features when we examine our integrated data in the study. So we see the time point information, but when we come here to customize our view, there are a lot of additional options. Now in this case, we're not particularly interested in the specimen ID information because these weren't specimens, and we're not interested in the visit ID because we used dates instead. But we are still interested in these comments. But now we might be interested in additional information, such as the gender of our participant and which treatment group they're part of. We may also want to view this data with particular lab test information, such as the viral load or the CD4 count. And then we could save this view for later. In this case, I'm simply going to make it our default view. And now, we're able to see that one of our participants, our female participant, has viral load measurements and CD4 cell counts, whereas our male participant does not. which in this case makes sense because our male participant is HIV negative, whereas our female participant is HIV positive. Now, just like in the assay area, we can chart this information. We can chart this data using any number of these options. But in this case, I'd like to show you a previously created plot, again, to save a little bit of time, which I have conveniently displayed as a web part on our main page. I'd like to look at CD4 levels per participant. Now we can see this time chart, as well as all of the other participants that in some way, shape, or form have been brought into this study. But what we're interested in is showing our new data, and you have the ability to add measures to this chart. In this case, we see all of the different data set options, but we are interested in our FCS protocol data. Here we have the gate 1 events that we might like to see. And you know, in order to avoid confusion, let's put this on the right axis. Now when it reloads, you'll notice this green line and this orange line that now appear integrated with our data. It's very convenient. These charts, they update live, so as you integrate more data or update the data sets that are there, these charts will automatically update, and you can save them for later viewing and export, as I showed you before. I'd like to thank you for your attention, and I hope that you've seen how LabKey Server can be used to further leverage the data from your flow cytometry experiments. Now I'd like to pass this presentation back to Kea, and we'd like to address questions. But actually, I'd like to go over this summary slide briefly first. So today, we talked about the direct export capabilities of FCS Express 5 into the LabKey server. We discussed report generation, as well as some of the analysis capabilities, and the web-based data sharing provided by LabKey. We showed how to design basic assays in the LabKey server, as well as how to export to LabKey using batch processing and FCS. And then how to view your data and some of the various data management and reporting tools available. Now we'd really like to take questions.